If a population is at Hardy-Weinberg equilibrium, then we expect the allele and genotype frequencies to remain the same from generation to generation. There are certain assumptions that we're taking into consideration when we apply the Hardy-Weinberg theorem. So if we discuss those, first of all, in order to have the Hardy-Weinberg hold true, there has to be no mutations. Okay, keep in mind that if we had mutations occurring, then we would have new alleles entering the population all of the time. So we don't want that to be taking place if we're going to have true Hardy-Weinberg equilibrium. The second thing that would be true all the time is that we're going to have random mating. We expect to have random mating, meaning there's not any individuals picking their mates specifically. They are equally likely to meet and mate with anyone of the opposite sex. Number 3, the third assumption is that there's going to be absolutely no natural selection. The fourth one is that we're going to have a very large population. And number 5 is going to be that there is no gene flow. And we'll talk about what we mean specifically by gene flow, but there's going to be no new genes coming into the population, and there's going to be no genes going out of the population. Now, it's fairly unlikely that every one of these assumptions is going to hold true for a population. Okay, so what this means, if they're not holding true, is that there is going to be some evolving or some evolution taking place. Now, let's take a look at an example. Suppose that we have a population of 100,000 human babies in a specific place. We've got a certain number of babies that have totally sickled red blood cells. So in other words, they have sickle cell anemia. We've got some that have partially sickled red blood cells, and we have another set that have completely normal red blood cells. Okay, so what we're looking at here is sickle cell anemia. This is something that is caused by a recessive allele. So let's go ahead and just assign our genotypes to start with. Okay, so these individuals here at the top, these have totally sickled red blood cells. So these are going to be our little a individuals. The ones with the partially sickled is going to be heterozygous, and the ones that have the normal cells, that's going to be big A, big A. Okay, so if we want to figure out if this population is at Hardy-Weinberg equilibrium, the first thing we need to do is we need to figure out what P and Q values are. So to do that, let's take a look at the babies that have the sickle cell anemia. So little a, that represents Q squared. And we know here that we've got 20,460 babies out of 100,000 that have little a. Okay, so to figure out Q squared, we would want to figure out what that percentage is. So go ahead and figure out what percentage we are dealing with here. So for Q squared, we've got 0.20 or 20% of our population has sickle cell anemia. If we take that information and we use it to solve for Q, remember that if Q squared equals this, then all we have to do to figure out Q would be to take the square root of both sides. So Q is going to then be equal to 0.45. That would be our Q. Okay, so if we know Q, we can very easily figure out what P is. So remember that P plus Q is supposed to be equal to 1. So if we know that Q is equal to 0.45, then P must be equal to 0.55. Okay, so now we know what P is. We'll just write it down here. We know what Q is 0.45. And now we can figure out whether we have a population at Hardy-Weinberg equilibrium. Okay, so to do this, let's see if we have the predicted frequencies that we would expect for the other genotypes. So for P squared is going to represent the big A, big A. Okay, so let's take P and that's going to be 0.55. Let's square it and see what percentage we come up with. Okay, so for P, we've got 0.30, or P squared is going to be that. If we figure out what the heterozygous ones are, that would be 2PQ. So let's plug in the information. We've got 2 times P times Q. Go ahead and multiply that out. Okay, so for Q, or sorry, for 2PQ, which is the heterozygous ones, we've got about 0.5. Okay, and so if we look at all of this, let's go back to the babies that we've got here. So look at the heterozygous ones. Heterozygous, we've got this many. Divide that out and see what percentage of heterozygous individuals we actually have. So 290 over 100,000. What's the frequency of that? Okay, so that comes out, really it's just a matter of moving the 0. We're going to have 0.59 is what we get for the heterozygous ones. Okay, if you compare that to what we predicted, okay, here's the comparison. It's off. It's not complete Hardy-Weinberg equilibrium here, because we can see that given the allele frequencies that we have, we're not getting the expected genotype frequencies. Okay, and one of the reasons for this is going to be that the individuals that have sickle cell, they're not going to be as equally likely to reproduce as individuals that don't have it. They are sickly. Some of them do not live a full lifespan. So that would be some of the explanation for what we're seeing here. Okay, so we do have some of those conditions that we had on the previous slide that are not being met. So if this is the case, as we had in that last slide, what we have is microevolution. So when we have microevolution, that is telling us that the population is changing. And when we say that the population is changing, the genotype and allele frequencies are changing. So how might microevolution actually occur? Well, there's a number of ways that this could take place where we could get a changing population. We could get changes in the allele frequencies and the genotype frequencies. So we're going to take a look at each one of these things. But one of the ways is going to be what we call genetic drift. Another possibility is that we have natural selection. We could also have what we call gene flow. We could have non-random mating, which is often the case. And we could also have some mutation taking place. Basically what we're doing here is we're contradicting those assumptions that we made with Hardy-Weinberg. So if any of these are taking place, then we do expect to have some microevolution taking place or changes in the population. So let's start to take a look at some of these things. What do we mean by genetic drift? Well, genetic drift, if you take a look at the picture here, we've got some beetles. We've got some green ones. We've got some brown ones or some tan ones here. And we've got someone walking along. They happen to step on some beetles. Okay, so their color has absolutely nothing to do with whether or not they get stepped on and killed by the man walking along on the sidewalk. But notice what happens. So he's walking along, he steps on a bunch of green beetles, and now you just don't have as many green beetles in the population. So now we would expect that there's suddenly going to be a lot more brown beetles around. So with genetic drift, we're basically talking about random events that can change the genotype or the allele frequencies. Okay, the key here though is that these are going to be random things that take place. There's a number of ways that this can happen. It's not obviously always going to be by somebody walking along and stepping on insects on the sidewalk. So if we take a look at some of the more popular examples of how we can get genetic drift, the first one is what we call the founder effect. So the founder effect is something that takes place when you have a few individuals that found a new location. So imagine that you have an isolated population, perhaps on an island, and a few individuals happen to make it off of that island and they suddenly colonize a nearby island. Well, whatever individuals those happen to be that got off of the first original island, they're going to help determine the genetic makeup of that new island that they're inhabiting. So let's take a look at that here. We've got this initial population here. It's made up primarily of darker blue individuals. And you've got a few of them that make their way to some new location. It's really random which ones happen to make their way to that new location. Then we have some generations that take place where they are reproducing. And then you have a new genetic makeup here. So we've got different allele frequencies in the new population compared to the old one that we had. Sometimes this is called bottleneck. But when we take the term bottleneck, bottleneck is really referring to the fact that there may be some kind of a disaster taking place. So with bottleneck, there's a disaster that pretty much randomly eliminates many individuals. Okay, so if we had a flood, we would expect a lot of individuals to be killed. If you had an earthquake, a hurricane, something along those lines, we would expect to have many individuals just randomly killed off. Here's a look at this. So a lot of times this is represented by beads or marbles inside of a bottle. And that's where the name bottleneck comes from. So notice that the neck or the opening in the bottle is really small. So some kind of random event is going to make it so that only a few individuals can survive. That's all. So if you were to take that bottle, tip it upside down, it's going to be random which marbles actually fall out. And that's representing the fact that it's going to be random which individuals survive whatever occurrence this is. So this is going to be some kind of a disaster that's taking place here. Okay, so a few survive. Those surviving individuals are going to reproduce with one another. And then when you look at the next generation, it's very possible that you're going to have different allele frequencies than you had in the original population before the disaster took place. So notice that originally we had a lot of yellow marbles. After the bottleneck, we have a lot of blue marbles. So one key here is that for genetic drift to take place, a lot of times what we're doing is we're reducing the population. So we're reducing the population size at some point, and when we reduce the population size, this is going to lead oftentimes to a decrease in genetic diversity. These are things that happen just because of the fact that you're taking suddenly just a few individuals from your population and they are the founders, or they are the ones that are going to reproduce and produce the rest of this population. Here's just a couple of examples that we have in the world today. The cheetah population is very, very small in the wild. Because we have so few cheetahs, they are basically interbreeding with one another. But there's not a lot of gene flow because they're so related to each other because you don't have very many individuals. So we have a lot of interbreeding taking place. Another example are these seals here that we have in California. Again, small populations are going to lead to a lot of interbreeding. These are disasters, whether they're man-made disasters that cause us to have small populations or natural disasters that cause it. In the case of the seals, it's going to be man-made, and a lot of times the cheetahs as well. Either way, we are reducing the genetic diversity because we're reducing the number of individuals. There's not going to be as many different alleles to be passing around or trading with one another once you eliminate individuals from the population. All right, so the consequences of genetic drift, we've already mentioned these, but the main consequence is that there is going to be a decrease in genetic diversity. We'll see that this has a direct impact on natural selection. So what this is going to do is it's going to lead to a decrease in the ability of the population to adapt. So this is going to work against natural selection. And we're going to talk more about natural selection. So if we look at these two things, we have a decrease in genetic diversity and we have a decrease in the ability of the population to adapt. Obviously, these are bad things. So genetic drift is not considered to be an ideal situation for a population, for a species. Gene flow is another example where we can have some microevolution taking place. As this picture represents here, with gene flow, we basically have a flow of genes or flow of alleles from one population to another. Here we see a brown beetle population and we have a green beetle population. And if somehow one of those beetles manages to make its way into this population, which was entirely green beetles before, we have now the introduction of a new allele. So that's what gene flow can do. It can introduce new alleles that weren't there before to a population. This can reduce the differences between two populations. So if you look at these pictures, you can imagine that if we had some tan beetles moving one direction and we've got green beetles moving this way, that eventually the two populations just look like a mix of brown and green. That's fine, but we don't have the differences that we had before. We're still going to have all of our alleles present. There's still going to be a lot of diversity there because we're not just killing off a bunch of individuals. There's going to probably be some random mating taking place. So we're just reducing the overall differences that we would see between the two populations. If you think about if you took two purebred dogs, different breeds, you mated them together, you're going to see a mixture of the two. If you continue to do that, then we just get dogs that begin to look similar to each other. If we're not keeping those two different breeds or 10 different breeds, if we're not keeping them separate from each other, all of the dogs would begin to look the same. We talk about natural selection. Natural selection is a big one here because natural selection is basically unequal success in two things: survival and this "and" is a big part of it, reproduction. So not only do we have unequal success in them surviving, so they live, they are also able to reproduce some offspring. So the idea here is that they're going to reproduce offspring, which is going to allow them to pass genes or alleles to the next generation. Here we have some different beetles. Suppose that the green ones are really tasty, The brown ones, not so much. Okay, the birds are going to go in there. They're going to pick out the green beetles. What is this going to do? It's going to eliminate a lot of the green beetles, and this means that the brown ones are going to become more plentiful. Why? Because the brown ones have a better chance of survival, which is going to lead to them having a better chance of reproduction. So they're likely to have more offspring. So what do you expect to have in the next generation? We expect to see more brown ones. And then in the next generation, we expect to see even more brown ones and even more brown ones, until eventually we may even end up with a population which is entirely brown beetles. That's what we mean by natural selection. It's differential success or unequal success in survival and reproduction.